I'd like to, first of all, thank US Poultry and Egg for allowing me to come here and talk about my research. About 14 years ago, I put in a grant to look at blackhead disease in turkeys. And back then, blackhead wasn't a problem because we had some treatments that were available for blackhead disease. But US Poultry and Egg had the foresight actually to start funding research into flagellated protozoa, which now, as we know, are causing significant problems with both the turkey and the chicken industry. So blackhead disease, just a quick overview, is caused by the protozoan parasite Histomonas meleagritis. This is a single cell anaerobic protozoa that actually dies quite readily in the environment. But even though it's a fragile organism, it can cause up to 100% mortality in turkey flocks and even up to 30% mortality in chicken flocks. So birds with signs of the disease are like all signs of the disease. They're going to have hunched posture, they're going to huddle together, but with blackhead disease, you'll start to see sulfur-colored droppings in your litter. So the current diagnostics for this disease is what we call pathognomonic, which means that if you've seen blackhead once, you can diagnose blackhead for the rest of your career. So blackhead starts out in the Sica, and what you get is ulceration and inflammation of the Sica. From the Sica, the parasite will migrate through the portal vein to the liver, and in the liver, the parasite eats the liver and causes major necrosis of the liver. So it's this destruction of the liver that results in the death of the animal, as well as, since the liver's not functioning, that's why you get those yellow droppings that are there. But as we talk about blackhead disease and Histomonas, there's always a partner in crime. And the partner in crime is Heterachus gallinarum, which is a cecal worm that's found in a number of different hosts, especially gallinaceous birds. Heterachus has low pathogenicity, so most people really aren't too worried about Heterachus being in their chicken flocks. The problem with Heterachus is it is the only nematode to carry Histomonas. So Histomonas will be carried by the nematode eggs. And so the life cycle is one which we call a direct life cycle. So you have a chicken that's infected with Heterachus. And just to give you a kind of idea, this is the number of Heterachus worms that we took out of 3 broiler breeders. So there are thousands and thousands of these Heterachus worms that can be found in the Sica. Those worms are then going to release their eggs, which will come out in the feces, and the eggs that are in the feces, remember, have Histomonas. So now you have an egg that's highly stable for years, carrying the pathogen that you're worried about. So it takes about 2 weeks for these eggs to undergo embryonation, or in other words, to be infective. And then as the bird eats those eggs, it becomes reinfected with the Heterachus larva. So you can see as birds are longer and longer lived, there's going to be more and more Heterachus larvae that are going to show up in their Sica. One of the other problems is the Heterachus eggs are really sticky. So they stick to about everything. And so insects, equipment, boots, darkling beetles, they can all act as these mechanical vectors and drag these eggs to other poultry units that are in the vicinity. So if you have a chicken unit that's highly contaminated, those can be dragged into a turkey unit, or even backyard flocks, or backyard flocks dragging it into your unit. So how do we get rid of these Heterachus eggs? Well, that was one of the questions that we asked and had funding from US Poultry and Egg. And in the literature, it showed us that if you have high temperatures, you can kill the eggs and the embryos or the larvae in them, but really high temperatures. But you can use composting to do that. And if you have low temperatures, the eggs won't develop. But in the industry is pouring a lot of salt in buildings to try to make sure Heterachus doesn't cause a problem. Limestone is being applied both inside and outside of buildings. And then there are a number of commercial products that have been suggested actually to be able to work to eliminate your Heterachus egg problem in your facility. So we just asked a simple question: can we develop an in vitro assay so we can start testing whether or not this is the case? And my graduate student, Catherine Cupo, came up with kind of an elegant experiment. And that is, we would collect eggs, she would put the product on the eggs, and then come back and add a dye called trypan blue. So if the egg is damaged, the dye can get in, and you'll see that the egg turns blue. And if the egg is not damaged, then the egg stays clear and white. So any eggs that turn blue, we would then take and we would desiccate for 2 days. So we dried them out so that the larva could die if it got desiccated. And then we just broke them open and asked the question, did they move, right? Are they alive? So 2 stages. Does the egg turn blue because it has damage? And 1, does the larva die because of that damage? And so this is a lot of research all together. And really what we found was high levels of bleach can damage the egg and kill the larva. Low levels of bleach can cause the egg to be damaged but not kill the larva. And then many of the products that are being tested, salt, limestone, diesel fuel, 6 hours in diesel fuel does not damage a heterachous egg. So any old timer that tells you that happens is actually not telling the truth. But even many of the products, actually all the products we tested, had a difficulty in damaging this egg. So really what the take-home message is that we need to identify products, maybe that can be combined with a chlorine ion to be able to damage these eggs. But the other thing is you need to get them out of your house by really cleaning those houses out. So since chicken houses, especially long-lived chicken houses, have heterachous in them, and we know the heterachous has histomonas, what the reality is almost all broiler breeders are going to be infected at some time with blackhead disease. And so our lab asked the question, okay, from what we're seeing in the field with blackhead causing a problem with broiler breeders, do we see the same thing in a laboratory setting? So we took a flock that we were raising at NC State and we infected them with histomonas on day 25. And then we collected data for mortality, body weight, uniformity, and also egg production all the way through the 64 weeks. So just as a side note, turkey guys don't like doing broiler breeder trials because they take way too long. So here's the data. So we're looking at days post-infection. And so what you can see here is that you have the control birds and the infected birds. And although the infected birds had a decrease in body weight, this was actually didn't reach a p-value of 0.05. So there was some reduction, but really not a statistical reduction. It wasn't until actually at the end of the flock, 420 days later, that we noticed that the infected birds were significantly lighter than the uninfected birds, like a long-term consequence. If you look at the Sica score 5 days after, out of a 0 to 4 scale, the birds started getting sick, as you'd expect they would. 10 days later, they had scores of 3, so that the Sica were readily involved. But by 20 days, the birds had already recovered or started to recover from the disease. And we saw very few liver lesions in the chicken. So if birds are infected, right, they got the disease, and in fact, when we sampled these birds at the end of their life, they were still positive for the parasite. So the parasite just remains part of their microbiota. So what about egg production? Well, I can tell you there was no difference in egg quality, onset of lay, and our infected birds actually outperformed our uninfected birds. So why did that happen? It's possible the increased egg production we got was mostly because our broiler breeders were a little lighter, which is also associated with egg production. But it brings up a real question of whether or not blackhead is really the primary problem when we're seeing this disease pop up in the field. We've done previous research showing that layer flocks also recover from the disease with no impacts on their production. We know that the chicken can mount an effective immune response. And so what is brought to our lab's understanding, as well as the things that we're testing, is really when you're seeing blackhead in a chicken, this is due to a secondary problem. You have an environmental or a disease stress that is reducing the immune system of that bird. And when the immune system is reduced, then that bird is then able to get blackhead disease at a high morbidity and start to get mortality. So if you have a flock that is a broiler breeders that's breaking with blackhead, I would suggest you go back and say, do we have ammonia? Do we have mycotoxins in the area? How is our nutritional program? Is cocci really bad, or are we looking at IBD and Merrick's or even E. coli that's been known to be associated with outbreaks of blackhead disease? So this causes 2 problems. Since chickens really can recover from blackhead disease, and they also are infected with heterachous, they spread a lot of worms into the environment. And so these worms in the environment can be taken by these mechanical vectors we talked about and brought into a turkey unit. And turkeys have a little bit of a disadvantage when it comes to blackhead disease. So as these eggs are brought into the turkey house, a turkey will eat a heterachus egg, and it will become infected with heterachus and with histomonas. But within 14 to 18 days, that turkey that took in that egg will die from blackhead disease. So heterachus doesn't have time to go through its life cycle. So if you look at a turkey flock and try to find heterachus larvae in their Sica, rarely will you find them because the birds have died from blackhead disease. So histomonas gets transferred a little bit differently in the turkey, and that is that as the turkeys huddle together when they're sick, they start to spread the disease laterally through fecal-fecal direction. So from the feces of one bird gets pulled up through the cloaca of another bird. And we know we can block this. We just put a fence in between the birds of the uninfected and the infected, and we won't get disease transmission across the flock or in the pens that we do our research with. So how does this work? This works through this little process that birds have. Is called reverse peristalsis. So when a liquid hits that cloaca, that bird, just like it would when it's being fertilized, will pull that material into the into the cloaca, and then through reverse peristalsis, this area where the birds are able to uptake water to maintain their water balance, pull that into the Sica. So now histomonas gets in the Sica, starts the infection, causes the infection that we saw in the Sica, and then can transmit through the portal vein to the liver, where it causes much more destruction and the death of the bird. So this is the method of transmission. So it makes for an easy model, right? And I use the word easy because nothing's easy in science, but if we have 10 infected birds, put them together with 10 sentinel birds, we can ask the question, does a product block the transmission of the disease from the infected birds to the uninfected birds? And this is what histostat 50 or nitarsone did. It didn't protect the birds that were infected, it just blocked the transmission of the disease from the infected birds by reducing histomonas counts in the Sica. So that's what we're looking for. So when I got to NC State about 4 years ago, we ran one of these big trials for a company that was looking at different essential oils that may play a role in the block of transmission. We used the same model that we had used at the University of Georgia, and we ran this trial. So you can see this is the percent of birds that are infected. In black is directly infected by the researcher, and in red is going to be the ones that are laterally infected. So the control, we had about 50% of the birds get directly infected. Interestingly and concerning, as we added essential oils to the diet, in some cases we got an increased rate of direct infection. But in no instances did we have lateral transmission of the disease. Right? The model I just told you that works wasn't working for me when I got to NC State. So I panicked, told the company I was super sorry that our model didn't work, and we repeated the trial, reduced the cage size, the pen size, so we'd have increased density, and we repeated the trial again, and our results were identical. So I'm sitting here trying to think, how is it that my model is failing? And it took me a trip to the beach to realize that I'd cured blackhead disease, right? Because we don't have lateral transmission. So we had to go back and try to ask the question, what is it that we did different? Why is it at NC State we're not seeing our disease model work, but when I was running this model at Georgia, the disease model was working? And the problem was a nutritionist. It's always the nutritionist's fault. When I got to NC State, I had Peter Firket design me a turkey diet, and he did the best he could do, right? He made a diet that was gonna make the turkeys healthy. It was gonna make their droppings tight so that would reduce ammonia in our barns, and it was a great diet. And when we went and looked back at the blackhead diet that we had used, this diet was a terrible diet. It was probably designed by a molecular biologist, where it had low crude protein, amino acids are out of balance, the salt was nearly twice as high as it should be, low in calcium and phosphorus. I mean, this is just a bad diet. In fact, it makes the birds drink a lot of water, and it loosens up their fecal droppings. So what's the effect of that? Well, when you have a loose or a wet fecal dropping, it increases reverse peristalsis in the bird. So the bird is naturally going to pull up more material from its environment. That loose dropping, when it hits the cloaca, is more likely to be pulled up than a thick dropping. So what we found out by this experiment was really this transmission of the disease in the turkeys isn't quite as easy as putting two birds together. It really deals with birds being unhealthy, them having wet feces. And that opens up a whole, Pandora's box, right? Because coccidiosis can cause birds to huddle, and it also can cause wet feces as the birds are flushing. And we've seen in the field that when we have coccidiosis, we have higher rates of transmission of blackhead disease. As I showed you, if we screw up the diet, we can also get increases of rates of transmission. We're looking right now under stress and even alternative products. If you notice, some of those essential oils started creeping up our rates of of infection of the disease. So we ran a simple battery test just to ask if some of these questions we could actually prove, like our hypothesis was right. So we took Dr. Friquette's diet and we decided to mess with it, and then compare it to our old blackhead model. And so what we're looking at here is just lateral transmission that occurred. So uninfected birds, you'd expect no disease. The NC State diet, we saw no lateral transmission is what we saw in the pens. And then we started adding magnesium sulfate, right? Something that's gonna cause the birds to flush. All of a sudden we started seeing lateral transmission. When we added dextran sulfate, which is a product that damages the gut of the bird, causing them to slough their lining and begin to flush, rates of transmission also started to increase, as well as coarse corn, which is known to cause reverse peristalsis. We saw transmission. And then our blackhead diet, 20% of the birds within 1 week of the birds being infected were also infected. So what we had shown was that in the turkey, it's not just as easy as killing Histomonas or getting Histomonas out of the flock. It really deals with understanding the health of the bird and actually using more of a global approach to trying to treat blackhead disease. So this gives us a wide variety now of opportunities to look at. So one, we need to reduce heterachus in the environment. This is going to help the chicken people out because it's going to make sure that their birds aren't infected at an early age and their immune system is able to develop before they get infected with blackhead disease. That's also going to help out your turkey brethren who are scared to death that heterachus is going to get into their flock. At the other end, we need to improve gut health, both in the chicken and in the turkey. When a chicken is healthy, blackhead is not a problem. When a chicken is unhealthy, blackhead becomes a problem. So we can look at alternative medicines not just for their antiprotozoal activity, but look at alternative medicines as how they improve the health of the bird. We need to focus on those management things of keeping birds on feed, making sure birds aren't being chilled, making sure that we have clean water lines so that we don't get E. coli problems, make sure that their microbiome is healthy and they're not flushing, and then really focus on controlling those secondary pathogens. From research in our lab and also field research, coccidiosis is a trigger of blackhead disease. Those birds huddle, they flush, and they transmit the disease if it's in the house. So the last thing I just wanted to point out, and it's an idea that our lab is throwing around, and I think it's something that I'm trying to get others to think about, and that is this idea that the Sica is really a long ways down the digestive tract. So when we start looking at all of the problems that we're worried about, Campy, E. coli, Clostridia, Salmonella, they all live in the Sica, right? And we can clear them out of the small intestine, and the Sica is just going to reseed the small intestine for you. But the Sica is all the way down the digestive tract. So as we're looking at these alternative products to improve gut health, treat blackhead disease, treat coccidiosis, we really need to start asking the question, are we getting that product to the Sica? And if we're not getting that product to the Sica, we are not actually going to solve the problem. If we aren't getting that fixing the microbiome in the Sica, we can have as many anti-salmonella treatments as we want. We're still not going to solve the problem with salmonella or Campy or Histomonas. So it's kind of a little bit of a plea, push to US Poultry and Egg to really start looking at research where we can start to target the delivery of some of these products that we've shown in vitro work but we're unable to deliver to the site of infection. And with that, I'll just acknowledge the ones who do the research, that's the graduate students, and take any questions at this time.